Hello everyone, welcome back to Praveen Online Academy. Today we are going to discuss the chapter regarding reproduction. Chapter 6, Reproduction, the generating system. In this chapter we are going to discuss detail regarding the structure and process of reproduction in all living organisms. Generally we know that reproduction we classify into two categories, that is asexual reproduction as well as sexual reproduction. In coming to asexual reproduction, the only single parent involves in the formation of young ones. Single parent involves in the production of new babies, that is offsprings. Whereas in sexual reproduction, the fusion of male and female gametes takes place for the production of new individual. So we are going to discuss by the end of the chapter, you will get detailed information, the process of reproduction in both asexual and sexual reproduction. So coming to our chapter, so reproduction, the generating system. In plants and animals, reproduction is necessary for life process for continuation of life by producing offspring. Simply, reproduction means reproduction is a biological process, production of biological process giving new individual offspring. Simply, animals were giving birth to their new babies is known as reproduction. So given, do you think the reproduction occurs only for continuation of life? So due to the process of reproduction, the life has been continuous takes place. So along the reproduction, along the continuation of life, there should be a proper environment for living, growth, and as well as shelter. So along the life, if the living organisms should be survive properly, there should be a proper suitable environment and proper food, shelter to escape away from the predators. Along this process, the life has been continuous, it can be taken properly. So how does the organisms grow? How does they repair from the own parts of the place? So simply the organisms will grow by the multiplying cell divisions, multiple cell divisions that were occurring in mitosis. Simply the process of cell division, during the process of cell division, the living organisms were able to growing. Clear? Next, what again, how does the repair occurs? So due to the cell division by the process of mitosis, the reproduction, the organisms are able to grow by according to their ages. Next, what again, organisms are capable of giving rise to offsprings by the process of reproduction. Simply the meaning for reproduction is, what again, meaning for reproduction, organisms are capable of giving rise to offspring, simply new individual rise to offsprings by the process of reproduction. Some organisms may reproduce differently in different situations. For example, during under favorable conditions, paramecium, we given the example of paramecium, rise to more of this kind single parent by splitting into this happens rapidly and several of them form. So look here, so the process of reproduction, if the reproduction has to take place, there should be suitable and proper environment. During the favorable conditions only, the organisms will able to reproduce. So given examples of paramecium, what is given, paramecium rise to more often of a single parent. So how the paramecium is able to reproduce? Look here everyone, the paramecium is able to reproduce by the splitting into two halves. We know that paramecium, wait a second. So coming to paramecium, so we know that paramecium is a slipper-like structure. Slipper-like structure is a paramecium, so which is bearing the cilia. The cilia helps for the locomotion. So paramecium is able to reproduce by splitting their body into two pieces. From here, then the splitting of body takes place into two pieces. Then finally the division takes place like this. From here, the splitting has been takes place. The final separation will takes place. What is given by differently under favorable conditions, splitting into two. So splitting into two individuals. So in paramecium, the reproduction will take place by splitting into two individuals, and This two parent and new individual is able to live separately after splitting. So this can happen under the favorable environmental conditions. Clear? Next, during unfavorable condition, what happens when the favorable condition is possible? The reproduction can take place. But during unfavorable condition, what is given? Unfavorable condition to two paramecium come in contact and exchange their materials. So what is given? Two paramecium come in contact and exchange their materials. So during unfavorable conditions, two paramecium come in contact and exchange certain materials of their bodies and produce to form more tolerant to the tolerant. That is known as conjugation. Simply, conjugation means during favorable condition, the paramecium is able to split and give to able to reproduce. But during unfavorable condition, the two paramecium were coming close to each other. The exchange of nuclei will takes place. During the exchange of nuclei takes place, the survival method conditions improvement will be takes place. Next, what is given? Activity 1. The activity 1 regarding given one activity. So you have to take the card. Formation of bacterial colony in a milk. So this activity, first you have to take the two bowls. In two bowls, pour some amount of milk, 100 ml of milk in one bowl. In another bowl, pour 100 ml of milk. Before that, you have to so I am taking two bowls. So in two bowls, you are pouring 100 ml of milk. So the same bowl you have to pour 100 ml of milk. The difference is in bowl A and bowl B. In bowl A, you have to take the cold milk. The temperature is cold. The milk temperature is very cold. In the bowl B, you have to take the lukewarm milk. Clear? So I am taking two bowls. In one bowl, I am filling 100 ml of milk, which is cold, with the temperature is very low, that is cold temperature, and another milk that is 100 ml. I am taking warm water, lukewarm milk. So after that, you can add tablespoons of some amount of curd. You have to add the same quantity of curd into the two beakers. After adding that, wait for 4 to 6 hours. After wait for 4 to 6 hours without disturbing the glass tumblers. So for each hour you have to observe whether the formation of curd coagulation is taking place or not. So after 4 to 6 hours we can see that the formation of curd takes faster in warmer milk compared to the cold milk. So I am adding same quantity, that is 100 ml, and here also same quantity, that is 100 ml. But here we are seeing within 4 to 6 hours we are seeing that the curd is able to forming properly as compared to the cold milk, because for the formation of lactobacillus, the temperature should be 35 to 40 degrees Celsius. So here the temperature is very suitable. So during the suitable temperature, the formation of curd takes place rapidly. So here the cell division, the replication, reproduction, splitting of cells takes place into very rapidly under suitable environmental condition. From this experiment, we can say that during favorable conditions, the replication takes place more rapidly. Here it will take longer time, above the 6 to 10 hours. The temperature is less than the 20 degrees Celsius. So here conversion of lactobacillus. The lactobacillus is responsible for the formation of curd. Simply, there is a bacteria present in curd that we call as a lactobacillus. Clear? That is about regarding the activity 1. Next, coming to the activity 2. We given the asexual reproduction. So the examples. Okay, next coming to the asexual mode of reproduction. So generally we know that the reproduction were classified into two categories, that is asexual and sexual. Reproduction were classified into two categories, that is asexual and the other is sexual reproduction. So in asexual reproduction, single individual takes place for the formation of offspring, for the production of offspring. So there is no fusion of male and female gamete, that is ovum from the female, egg, that is ovum, egg from the female, and sperm from the male. There is no fusion of gametes takes place in asexual reproduction. But in the sexual reproduction, the fusion of gametes, that is male and female gamete, takes place, that leads to the zygote and formation of fertilization will takes place. Here you can male and female gametes takes place. This is the major difference between the asexual and sexual reproduction. Now coming to the asexual reproduction, let us see the structure, detailed structure. We are going to see the detailed structure regarding the asexual reproduction. So what is given? Asexual mode of reproduction. So organisms can produce asexually in many ways. Some of them are discussed below. Simply, there are many asexual reproduction are divided into many categories. So we'll discuss one by one. First, what again? Fission. Single cell organism. These are the single cell organism such as paramecium. Example of fission is given the paramecium. So and the bacteria reproduce by splitting into two or more offsprings. From here, look here. We know that paramecium structure generally look like slipper-like structure, right? So during reproduction, the paramecium is able to split. The process splitting takes place, and no individual will separate from the parent body. Single organs such as paramecium and bacteria, how they are reproducing by splitting into two or more offsprings. It may split into two or more than two offsprings. Clear? By transverse binary fission, the splitting will take place by the transverse binary fission, to dividing into two individual offs, binary two individual offs. That is known as the fission. Simply, fission means splitting into two offs for the production. That is known as fission. The example of fission is paramecium and bacteria. Next, coming to the budding. Coming to the structure of budding. A growth on the body. Look here. This is a budding. Example of budding. What is given? In the budding, a growth on the body as a bud that grows from nearly identical copy of the parent. When the bud totally grows, then it separates from the parent and survives independently. Look here, budding. So imagine is a parent body. So from the side of the parent body, a small bud appears to grow. This after a small bud appears to grow. So the size will increases. After increasing proper size, this body is able to separate, detach from the parent body. Look here, I'm explaining 1 second. It is a bud. So these are the buds. At side of that, there is a growing. It is a parent body, and its side a body is able to forming after proper size. The separation, the detach can takes place from the parent body. So again, these small buds are able to grow by their own, survive by their own after detach, after separating. That is known as budding. Clear? Example, we can see the process of budding in the yeast. Clear, everyone? Next, coming to the fermentation. Some of the organisms can grow fermentation, simply splitting into, look here, fermentation. Okay, let us read this sentence. After I can explain this clearly. Some of these organisms can grow from single piece of the parent organism. This can be from any part of the body. Fragmentation occurs only in simplest, such as some flatworms, molds, lichens, spirogyra. These are the examples. They may also reproduce sexually. Fragmented in the common mode of reproduction in algae, fungi, and many plants. Simply what I given. This can be from any part of the body. Fragmentation. It is a parent body. This can be formed at any part of the body. It may split here, it may split here, or it may split at any part of the body. After splitting, this individual is able to grow by their own. So after splitting, the separation takes place from the mother body, parent body, that is parent body, and no individual is able to survive. That is known as fragmentation. Look at this image, is a parent body. After that, the parent body is able to divide into two pieces, and again that another it is again is able to divide into four pieces. The rapid replication has been seen in the fragmentation. Examples of fragmentation. So what is given? Simply such as flatworms, moles, lichens, and spirogyra. These were the examples of flatworms. So whatever reproduction in the moles, lichens, and flatworms, the reproduction can take place by the splitting into pieces. And given they can reproduce by sexually. The examples of sexual reproduction is fungi and algae. Next, coming to the parthenogenesis. Parthenogenesis. So we know that asexual reproduction can take place by the single parent, that is without fusion of male and female gamete. But in the sexual reproduction where the fusion of male and female gamete takes place, so parthenogenesis in general sexual reproduction can take place by the fusion of applied gametes forming diploid zygote. Okay, but here the process occurs commonly in lower organs such as algae and fungi. Example given as a spirogyra. The process of development, look here, the statement from here you have to observe carefully. The process of development of young ones from unfertilized gametes is known as parthenogenesis. What is given? Unfertilized gametes. So simply the reproduction or the process of common occur organisms such as algae and fungi. Give an example. The process of development of young ones from the unfertilized gametes. So simply the new individual, the new offspring is able to forming without from the gametes. Without means unfertilized, undevelopment gametes is known as a parthenogenesis. In Greek word partho means virgin and genesis means production. Simply the production takes place from the unfertilized egg is known as the parthenogenesis. Example given the watermelon. So for the watermelon can take place by the process of parthenogenesis, and we can see the seedless watermelon, seedless fruits. Next, parthenocarpy. So nowadays we are able to develop seedless fruits like watermelon, grapes naturally. In some plants like banana, ovary direct develop into fruits without the process of fertilization. This phenomenon is called parthenocarpy. Look here. Naturally in some plants like what is given examples given as banana. So the ovary direct develop into fruit without formation of fertilization. So in banana, so for the formation of banana, whatever the structure of the ovary, that ovary is able to directly develop into fruit, that is banana, without fertilization. That phenomenon we call as a parthenocarpy. So simply in the parthenocarpy, the ovary develops into fruit. Whatever the ovary that directly develops into fruit without fertilization, means without fusion of male and female. Simply without fusion, the parthenocarpy is able to take place. The example of parthenocarpy is banana. So whatever the banana that you are eating as a fruit, it is developed directly from the ovary of the banana plant. Next, coming to the vegetative propagation, and before that given regeneration. So what is meant by regeneration? Regeneration means growing again. Abdikar, look here. Regeneration. For example, is a plant, it is organisms. So by whenever accidentally some part has been cutted, so after this part is able to regenerate again after some time. After cutting, this part is able to regrowth again after cutting is known as regeneration. Means again growing. Their original structure, original shape is known as regeneration. What is given? Many organisms have able to rise to new individual organisms from their body parts. That is, if the individual is somehow cut and broken. What is given here? Look here. If the individual is by the accident, it is cut or broken into many pieces. These pieces grow into separate individual and this similar to the fragmentation. Simply, for example, in the fragmentation, so in fragmentation, so same the structure look like a fragmentation. Imagine as an individual. So when the individual accidentally cut and this piece is able to separate, right, and this is able to separate. So from here the remaining body posture is able to generate after some time. For example, the structure is like this. If the small part is able to splitted, cutted, this small part is able to develop into new full structure. That is known as the regeneration. By the process of regeneration, and the new individual takes place. This process is quite similar to the fragmentation. Simply look here, is a body, the head has been separated and tail has been separated. After some time, look here, the head has been, tail has been separated. Again, after the separation, head, the full body is able to growing, and to the tail, the full head and full body structure is able to forming. That is known as regeneration, means growing the original body structure again. That is known as regeneration. Vegetative propagation. So vegetative propagation is a asexual reproduction, asexual mode of process that can be seen in plants. Okay. Vegetative propagation in higher plants, vegetative propagation may be natural or artificial. So simply natural means without involvement of human activities, that is known as natural. Without involvement is natural, and with involvement of human activities is artificial. So simply vegetative is also known as asexual reproduction. So whatever the reproduction that you are going to see in plants is known as vegetative propagation. So given natural propagation, so without involvement of human activities, what are the natural? So leaves and stems. Look at the example, is just structure of the bryophyllum leaf, and on the surface, on the corners of the leaf, we are able to seeing that new small individual plants were able to growing, right? Look here, look carefully here, the new individual plants are able to growing on the surface area of the leaf. So on the surface area of the leaf, new individual plants are able to rising, right? Is a bryophyllum plant. These plants were majorly found at moist atmospheric conditions, moist and water surface water, more abundant of water regions. We can see these bryophyllum plants. So leaves in bryophyllum, small plants can grow at the edge of the leaf. Simply, the small plants can arises from the edge of the leaves. So after shedding their leaves, that leaf will fall on the ground, right? So with the help of the moist conditions, these small plants are able to grow as a new plants. So simply in the bryophyllum, the small plants are able to rises at the surfaces, at the corners of the plant leaf. So after shedding the leaves will contact with the ground. After contacting with the moist with suitable environment conditions, this new plant rises, growing takes place. That is known as bryophyllum. And next, coming to the stems. Aerial weak stems like runner and stolons. The examples are runner and stolons. When they touch the ground, give off advantageous roots, and when the connection of the parents is broken. So look here. In the stems, take the examples. These are the leaves. These are the example as a grass. So when these leaves touch the ground, when this image is the ground, so when the leaf touch the ground, so this start growing, forming of new roots. So when the new roots were completely developed, these will be separate from the parent plant body. After separation, again the formation of new plant has been takes place. That is known as stems. In stems, examples runners and stolons. Simply, I am repeating once again. Look here. So is a parent body. So when the leaves were touching on the ground, so from here new roots were forming, new roots were arises. So after completely formation of new roots, this plant, the separation from the parent will take place and new individual, new plant as able to rises. Clear? That is the right examples, runners and stolons. We can see in the grasses. Okay, next what is given? Some examples of propagation by stem formation of stolons, bulbs, cones and tuber. These were the examples for the stem formation. From the stem, the reproduction, the replication can take place. Next, stolons. Examples again: jasmine, strawberry, and bulbs, that is onion and cones. Look at these images. It's a bulb. Okay, these are the root structures. Bulb. Look at here. Why I'm calling bulb means here the onion is able to bulge inside the ground. So bulbs, tubers, stolon, cones, and roots in parts in millingotine. Okay, these are the examples. Okay, next coming to the artificial propagation. So I said you that in the artificial there is the involvement of human activities, that is known as artificial propagation. Whatever the bryophyllum and runners, stolons, tubers, these were the examples for the artificial. Now examples for the natural. Now coming to the artificial with the involvement of human activities. So first given as a cutting. So look at this plant, you can get the idea regarding the cutting. Simply separating the stem of a plant and placing that stem in a potted. Simply separating the stem, any part of the stem of a plant, and placing inside the ground soil, that is known as the cutting. So simply he taken the separate of the stem, again the stem is able to placing in the soil. So after some days, after few days, the roots arises and completely new plants able to rise fastly. But again, some plants can grow individually when piece of the parent plant having bud is cut off. Some piece of parent body is cut off. Look here. What is given? Some plants can grow individually when a piece of parent plant having bud cut off and from the existing plant, the lower part of this cutting is buried in the moist soil. This is important. After cutting, after the separation of this part, and it is able to buried in the moist soil, which is having the moist soil, the soil is able to contain proper amount of water as well as the nutrients. Then after few days what happened? The cutted parts having the buds grow as a new individual, developing a roots. Example, rose and hibiscus. We can see in hibiscus and roses simply in the cutting. So we have to separate, we have to cut the stem. After cutting the stem, we have to bury it at the soil, in the soil with suitable conditions like proper availability of water and minerals. After that, after few days, the new roots were arises. After arising, the plant is able to growing. So when I cut this example, I taken the stem. So when I buried in the soil, the new roots were arises. After arising, the plant is able to grow properly. Example is given as a rose and hibiscus. Rose and hibiscus. That is known as cutting. Next, layering. A branch of plant. Next, look at this layering. Look here. A branch of plant with at least one node is bent towards the ground and part of the it's covered with the moist soil, leaving the tip of the branch and exposed to the ground. After few days, the new roots from the part of the branch is buried and the soil buried branch is cut off. Simply look here. You taken the grass. So these were the leaves. When the leaves touches the ground, so from here immediately the new roots will start rising from the leaf. New roots. Whenever the leaves bend towards the ground, so at the ground the new touches to the ground. So here the roots developing has been takes place. After some time, it is detached from the parent body, completely separated from the parent body, and new plant is able to rises. That is known as layering. Next, what again? Grafting. This is also important. In grafting, you have to So look at the grafting. What are you given? Two plants were joined together such way that stems join and grow as a single plant. So in the grafting, two plants were combined. Two plants. What are you given? The two stems were joined, grow as a single plant. One which is attached to the soil called stalk, and cutters stem of another plant without roots is called as scion. So simply this we call as scion, and this is stalk. Okay, whatever the cutted stem we call as the scion, and the plant with the proper root we call as the stalk. Clear? So grafting is used to what is given how to both stalk and scion are tied with the help of the twin thread and covered by a polythene cover. Grafting is used to obtain the plant to desirable character. The major reason for doing grafting is to obtain the desirable characters, to get the proper characters. For example, one plant is able to survive for during high temperature, high conditions, right? And one plant is able to, look here, there were two plants. Imagine there were two plants, okay? One plant is can able to tolerate during high temperatures with can survive with less amount of water, and this plant is able to produce more amount of yield, more amount of fruits and flowers. So while I'm combining this together with help of the grafting, so these two characters can obtain to the single plant body. So simply we can obtain the desirable characters. For that reason, to obtain the desirable characters, we can use the process of grafting. Clear? In the grafting, so in grafting, stem has been cutting, they will cut the stem, and the stem is able to attach the plant. So they can be attached with the help of a thread, and they will cover with the polythene cover. After some time, after some days, the plant is able to attach properly, and the two characters from this plant and this that is sequin and stalk, the two characters can seen in a single plant. So for that reason, we can use the grafting. The majorly the grafting can seen in mango, citrus, apple and roses. We can commonly seen in mango, citrus, apple and roses. These are the examples of the grafting. Clear? Next, what is given? Next, by grafting, very young cycle shoot part of a plant can be made into flowers and produce the fruits earliest. If we have two varieties yields, so from the grafting we can get majorly the grafting can used for the high production of fruits, flowers, and for the major reason for high yielding with the proper sustain to the environmental conditions. And next is given tissue culture. Tissue culture. So majorly we use the tissue culture for the commercial purpose. The tissues culture, they are being replaced by the modern technology of artificial propagation in plants involving tissue culture. In this tissue culture, few plant cells or plants tissues are placed. In the tissue culture, they can take the plant cells. By that cells, they can grow more number of offsprings, more number of plant species. In tissue culture, few plant cells or plant tissues were placed in a growth medium with the plant hormones in it and grows into new plants. Thousands of plants can be grown in a shorter interval of time. So tissue culture, in tissue culture, the major advantage of tissue culture is we can produce more offsprings within a short period duration of time. We can produce more number of offsprings in a short duration of time. That is the use of tissue culture. Next, spore formation. Formation of spores. Next, coming to the spore formation. In generally, we may notice that thread-like structures and blackish color, powder-like substance rotate on fruits and blood sizes of food material. Simply thread-like structures in this bonded, it is called with this sporangia. Okay. Spore formation. So generally we can see this spore formation in rotten fruits, rotten bread slices and other food. Simply the spores can seen in spoilage of rotten things like food materials, fruits and bread. Majorly we can see in the thread-like structure, white color formation. So this is structure of the spores. So from this, here is structure of spores, and these we call as sporangium, and these are the spores. Look here. Majorly we can see in the rotten substances like bread, fruits and food. So these spores were covered within the sporangium. These were the spores. These were covered in the sporangium. After some time, after few days, it is able to split. Means it is able to breakage. During breakages, many spores were separated. Many spores were come outside of the environment and distributed to the surrounding area. After distributing, during the moist condition, the spores start growing to the new individual. So simply it is covered in the sporangium. So after the blasting, the spores where these spores after these spores were separated and distributed in the atmosphere, and during the favorable conditions like favorable suitable environmental conditions like moist area, water content, these are able to grow into new individual species. So he given an activity for the formation of spores. He given one lab activity. Let us see that activity. To examine the rhizopores or common mole under the microscope. So for this lab activity, everyone know that for the activity, the material we have to write the aim, material required. So for the material required, we have to take the piece of bread and polythene cover, water, microscope as well as safety gloves. I mean safety kit. So the metal liquids are piece of bread. Piece of bread, water, microscope, safety gloves. Okay. So the step is procedure. So first you have to take a piece of bread slide and sprinkle few amount of water on it. So you have to take the piece of bread slide, take the bread slide and sprinkle some amount of water droplets on the bread. After sprinkling, place that bread under the cover that bread with the polythene cover. Cover that bread properly with the polythene cover and tie that mouth tightly without escaping of atmospheric gases from inside to outside and entering from outside to inside. And one more thing, before tying this, you have to place some amount of atmosphere, means you have to place some amount of gases in the polythene cover. After that, place this polythene cover 2 to 3 days at under proper sunlight conditions. Then after 2 to 3 days, we can see that the formation of bread, rhizopores growing on the bread. Look here, these are the rhizopores that were growing on the bread. After 3 to 4 days, we can see that formation of rhizopora. These are the formation of rhizoporas were growing on the bread. So simply when you're taking bread, when you're sprinkling on water, some amount of water and packing that bread piece with a polythene cover. After 2 to 3 days, the growth of rhizoporas can seen clearly. So after that time, take some piece of bread and place it under the slide, and with help of water place it under the slide and cover the slide cover slip and place under the microscope. We can see the rhizopores through the microscope. Look here, the rhizoporous mycelium seen under the microscope. These are the visible. This is a photo of the microscopic view. So this we call as a sporangium. It's the spores. These are the spores. Sporangium spores.